Very good morning, children. Welcome back to the biology class. Today, our topic is stomatal transpiration. You are well aware of transpiration, the process of loss of water in the form of water vapors from the stomata of the leaf or leaves is transpiration. Whereas the transpiration is further classified into three categories based on the part which is actively involved in this process. For example, transpiration is taking place through stomata, then it is termed as stomatal transpiration. If the transpiration is taking place by cuticle, then it is termed as cuticular transpiration. If the transpiration is taking place by lenticles, then it is termed as lenticular transpiration. We will discuss about the other two modes of transpiration, that is cuticular and lenticular transpirations later. Before that, the number one transpiration, that is stomatal transpiration. We will discuss it now. See children, here this is a cross section of leaf. Cross section. A thin section of a leaf is taken. Transversely, a thin section of the leaf is cut and a slide is prepared. If the slide of cross section of leaf is placed under microscope, and when we observe it, it looks like this. When the thin section, transverse section of the leaf is observed under microscope, making a slide, we are able to see this. This is what we call an anatomy of a leaf, internal structure of a leaf. This is what an internal structure, anatomy of the leaf. See here, this is an upper epidermis and this is a lower epidermis. This is upper epidermis and this is a lower epidermis. And these cells we are able to see here. These cells which are arranged in a row without leaving any intercellular space. We don't find any intercellular gap, intercellular space between them. Is it? Whereas down below these cells, the other cells are arranged. They are not arranged as these are arranged in a row equally. They are arranged in a zigzag manner. Is it? So these cells we call palacyte parenchyma. What we call them? Palacyte parenchyma. The cells which are arranged in a row without leaving intercellular space or gap. Palacyte parenchyma. These are palacyte parenchyma. Whereas the down below these cells, which we can find some intercellular space between them, among them. So we call them spongy parenchyma. So when the internal structure of the leaf is taken, anatomy when we observe, we are able to see mainly palacyte parenchyma and spongy parenchyma. And here, what we are able to see, this we can call a vascular bundle. What we can call this? Vascular bundle. What is vascular bundle? An association of xylem and phloem tissues. Xylem cells are there and down below there is phloem. So the xylem cells and these are phloem. So the xylem are water conducting tissues and phloem are food conducting tissues in plant. So the arrangement of xylem and phloem together, a group, an association of xylem and phloem in a group, we term it as a vascular bundle. So we are able to see palacyte parenchyma in the cross section of the leaf, spongy parenchyma, and here we are able to see a vascular bundle which is composed of xylem and phloem, water conducting and food conducting tissue. In addition to this, we are able to see a huge air space. We can call this as air space. Air space. See, this air space is left by this spongy parenchyma. Therefore, we say spongy parenchyma are the cells, they leave intercellular gap. This air space we are able to see here due to the space gap left by spongy parenchyma. Whereas palacyte parenchyma do not leave any space here. So they are tightly packed, closely packed. So we say them palacyte parenchyma cells in a cross section of the leaf, They do not leave, they do not show any. There is no intercellular space among palaceid parenchyma. There is an intercellular space among these spongy parenchyma, so the air space we are able to see. Now here, what is the mechanism of this stomatal transpiration? And before that, palaceid parenchyma, they have more amount of chlorophyll, and the spongy parenchyma have less amount of chlorophyll. They take part actively during photosynthetic activity. They are less actively involved during photosynthesis. This is the difference, main difference between these two cells which we are able to see in the leaf. Palaceid parenchyma arranged in a row, they do not have intercellular space. Spongy parenchyma, they are not arranged in a row, they are arranged zigzag manner. They have intercellular space. Palaceid parenchyma cells have more amount of chlorophyll. Spongy parenchyma have less amount of chlorophyll. They are actively involved during photosynthesis process. Palaceid parenchyma. Spongy parenchyma less actively involved during photosynthesis. And in addition to this, palaceid parenchyma, spongy parenchyma, vascular bundle, and air space, we could able to see on this epidermis. This is an upper epidermis. Here we can say lower epidermis. On the lower epidermis, we are able to see the opening of stomata. Here, this is a guard cell. The stomata, as it's composed with a guard cell, a kidney bean-shaped guard cells are there. This shows a stomatal opening. This is the opening of stomata. So the mechanism of stomatal transpiration, what happens? Compared to cuticular and lenticular transpiration, most of the water is lost during stomatal transpiration. Means compared to lenticular and cuticular transpiration, the loss of water takes place through stomatal transpiration is maximum, higher compared to lenticular and stomatal transpiration. The loss of water, capacity of movement of water during stomatal transpiration is more when compared to these two, lenticular and cuticular transpiration. Stomatal transpiration has more loss of water during this process. So see here, what happens? Here we can see, this is number one, movement by osmosis. Water movement takes place by osmosis. And number two, the movement of water takes place through evaporation. Number one is movement of water by osmosis process. What is osmosis? The movement of ions or molecules from the point of higher concentration to lower concentration till the concentration of both the sides becomes equal. We can give this definition for osmosis. What is osmosis? I repeat, during osmosis process, the movement of ions or molecules of that particular substance in which the osmosis is taking place, they move from higher concentration to lower concentration. That means from more crowded area to lesser crowded area. Who moves? The ions or molecules of that substance in which the osmosis is taking place. So the movement by osmosis takes place first. During this, the ions moves from higher concentration to lower concentration. At that time, the force of attraction between the cell wall of cellulose, the pores of cell wall of cellulose, and this vascular bundle, water molecule, the force of attraction between the pores of cellulose of the cell wall of cellulose and water molecule increases. Due to the cohesive force increases, and therefore water comes in contact with stomatal surface, and there is a movement occurs by osmosis process. We call it as movement by osmosis. And the other one, the movement by evaporation. How the movement by evaporation takes place? In this, what happens? The water from lost from the cells get collected in the air space, and air space from the water. Once the air space are filled, there more water molecules are crowded here. They are water further pulled towards the stomatal opening, and it is given out. That we call movement of water through stomatal openings. In this way, what happens? The mechanism of stomatal transpiration occurs.